Hello everyone, this is Mohammed Elbaz from Northwestern University. Today I'm delighted to be presenting our work that's recently accepted in MICCAI 2021 entitled Stochastic for the Flow Vector Field Signatures, a new approach for comprehensive for the Flow MRI quantification. For the Flow MRI has recently been introduced as a novel imaging modality that allows direct quantification and direct measurement of non-invasive blood flow dynamics in terms of velocity vector field over the three over three spatial directions as well as three spatial dimensions and times. Therefore, it provides us with dynamic vector field mapping information, which can be seen here on this visualization of that vector field in a patient with bicuspid aortic valve disease. Patients with bicuspid aortic valve disease has been shown recently based on 40 Flow MRI studies to develop complex flow changes in throughout the aorta as a result of the valve abnormality that associate with the disease severity as well as the potential patient outcome. It's important to note, however, that the flow changes in these patients as well as other cohorts, not necessarily a simple flow changes. It can be, as we can see here, a spectrum of flow changes. For example, it involves multiple complex flow patterns. For example, here in this patient, we see a flow jet happening together with a vertical flow that's happening over here, as well as helical flow that's happening throughout the aorta. So this is a complex spectrum of flow changes. Unfortunately, however, current techniques are unable to directly quantify the velocity vector field information that's rich from 40 Flow MRI, and instead what the current methods does is they require to convert that comprehensive velocity vector field information first to some sort of a scalar field derived from that data. So basically becomes a secondary field, usually based on a flow dynamics operator such as kinetic energy, vorticity, or wall shear stress. While each of these is helpful, it does not fully capture that spectrum of complex flow changes. For example, as we can see here, this kinetic energy would capture the flow jet that we see on the velocity field. However, it will not be able to capture that vorticity, or the vortex flow. Vorticity would be vice versa. It captures the vorticity, but not the jet. Wall shear stress will capture the forces on the wall, but not what's happening within the aorta. Therefore, none of these methods are able to capture that complex spectrum of flow changes. Therefore, they might be, in fact, under utilizing and under assessing the severity of the disease based on the under assessment of the complex spectrum of flow changes in this case. Therefore, the purpose of this study was to introduce a novel stochastic 4D flow vector field signature technique that's able to directly, and perhaps this is the first method that able to quantify the vector field information in its entire dimensionality for the flow MRI. In a standardized as well as a linear complexity and robust reproducible way. Our methodology consists of two stages. We first construct that signature stochastically. Within the second stage is to introduce a hemodynamic signature index that's able to allow us to quantify the differences in these captured flow signatures. Our method, the input for our method is the segmented aorta, which basically the vector field information, the entire vector field information, three direction, three dimension, and over time within the aorta is what we see here. That's our input to the method. Our method, the first stage is we do multi-manual stochastic for the uniform random sampling over the entire domain X, which is basically all the indices over the entire time duration. And we do that using the sampling, the pairwise sampling. We do it systematically based on a function eta, which is a function of the patient-specific size of the aorta, and as well as a density factor that makes sure that we adjust the density and make it equivalent among different patients, even if they have different sizes of the aorta. Then it allows us to for systematic comparison. And in this case, this was we had about 6 million pairwise samples based on this function, the patient-specific function. The second stage is we then define what we call a co-association parametric function that works vector-wise to allow us to capture or quantify the pairwise vector association between each pair of vectors. Here we use the angular dissimilarity metric as our co-disparity function between each pair of vectors. That actually these vectors are the vectors that correspond to the pairwise sampled but fetched for the Flow MRI. And then it's important to know that this angular dissimilarity is intrinsic vector field that works directly on the vector field, does not require any conversion to scalar field information. After that, we define our signature as the probability density function of this multi-million thetas or angular dissimilarity matrix. It's important to note here that the theta here is it works as a dissimilarity function. So if the values of zeros means that they are completely matching vectors, and 180 means that they are completely dismatching vectors, and then we have the range of in between. So importantly, because of this is defined as a probability density function, is by definition standardized. The area under the curves equals 1, as well as a volume normalized, therefore allows us to systematically and consistently compare between different patients, even if they have different aorta sizes as well. The second stage is we compute the hemodynamic signature index. The hemodynamic signature index is defined based on which is basically a measure of a difference between the distribution of that signature that we defined of each patient compared to all controls, and then we take the 95 percentile, which will be each patient will have a corresponding HSI index. Same for controls, we define each control, the baseline control signatures also from this method similarly based on the earth mover's distance AMD, which is the distance between distributions. The output then will be two vectors correspond to the number of patients and controls, so we are able to compare them together. Our data set involved 106 scans, including 57 bicuspid aortic valve patients, 34 healthy controls, as well as 15 healthy controls underwent second scanning 2 weeks later to understand the reproducibility. We assess the convergence, the signature sensitivity, as well as the rescan reproducibility. Our results show convergence of that density, the patient-specific density metric, allowing us to do equivalent comparison between different patients and controls consistently. Our method also showed that average error to the randomized segmentation errors was actually in the signature was average was about 1%, the maximum was about 5%, which is in the worst iterations, which is actually extreme errors in the segmentations. Therefore showing the robustness of our method. Our method also showed there is a clear difference in the profiles of patients in red compared to the healthy controls in black, and that was characterized by increased level of higher values of thetas or angular dissimilarities, which correspond to the or the densities basically of these were higher in the patients, which basically correspond to more heterogeneous vectors in these patients compared to controls. We also were able to see that this was characterized also by hemodynamic signature index with higher levels of hemodynamic signature index in patients compared to controls. We should also here there is an excellent Scan-Rescan, almost perfect agreement in this signature comparison. Our method also has linear complexity, allowing for near real-time analysis because of the vectorized pairwise computation, about 3 seconds per signature computation. Our method is comprehensive, allowing the full utilization of the velocity vector field with no derivatives or scalar field conversion needed, making it inherently more robust, but also the first method that can fully characterize the full spectrum of fluid changes from the vector directly from the velocity vector field. It is standardized because of the probability density function, as well as registration free because it's a difference between distributions. It is a linear complexity, very fast, real time. It is also robust to the uncertainties because this will appear in the tails of the probability density function by definition if they are within small densities, and then it is also reproducible, flexible to any coordinate system, Cartesian, curvilinear, and so on, as well as provide a generic mathematical framework to quantify vector field data beyond velocity, as well potentially a tensor field or even higher dimensions. So in conclusion, we propose a novel data-driven methodological stochastic approach, which is the first method that can allow to directly quantify the velocity vector field information comprehensively over the entire field for the flow MRI, allowing us to understand or to quantify or capture the spectrum of complex flow changes. And with real-time processing in over 100 patients, we showed the convergence, the excellent robustness to errors, as well as excellent reproducibility, as well as importantly the diagnostic potential by identifying distinct flow signatures in bicuspid aortic valve patients versus controls. In the future, we will extend this technique to assess different cohorts of cardiovascular disease and association to clinical outcome. With this, I'd like to thank you all for your attention, and I'd like to thank our funding sources. Thank you very much.